Ooh, okay.
It's
crumbly.
That looks really altered.
Yeah,
Yeah.
that might not even be good for analysis.
Okay.
So that's okay. Thanks for all the efforts.
Yeah.
I think we can keep on moving. Yeah, these rocks look quite a bit more weathered than the ones downslope. At least they got one rock on this dive though, earlier before our watch.
Oh, nice.
Yeah, but in general we were kind of trying to target collecting one every 1,000 meters if there were good candidate rocks. Could then age date potentially different lava flows, different eruptions.
Let's zoom on that.
What is that? What is that? Push in.
It's
Anemone?
a Corallimorpharian. I like them because they have those little white balls on the ends of the tentacles.
All right.
Yep, that's great. Thanks.
Come wide.
Now, is there a reason for like those little balls on the ends?
Well, the Corallimorpharians always have the little balls on the ends. I don't know the reason for them now. But it makes them very distinctive. Bridge, Nav. Can we make a 30-meter move, 320? Thanks.
As we move up slope here, earlier when we were chatting about fishes and the depths that they can live at, we had some really nice comments from a scientist ashore, Kenneth Sulak, who mentioned that the determination of the deepest limit for fishes has actually already been exceeded. The Pseudoliparis is in some abundance has been observed scavenging at over 8,000 meters, at 800, 336 meters. So
Looks like you got something off to the right there.
it's quite interesting that the ocean is so big. We haven't observed all these depths that
Where it's like an
some of
optical
our-
illusion.
some of our ideas may.
Big stick.
It's
Yeah.
kind of hard to set like limits
It's a
and
piece
stuff,
of
isn't it,
wood.
when you
Yeah.
haven't looked at
Big
so
old
much
stick.
of it.
Yeah, let's look at the stick.
Exactly.
Sure.
Sometimes weird stuff lives on sticks. Oh, look at all these little squat lobsters.
lobsters.
It's a squat lobster party.
Is it a stick?
Yeah, it's definitely wood.
Nice. They, I think the watch after us at one
Push in
point
there.
saw coconut.
Yeah, I saw the coconut.
Oh,
All
yeah.
these squat lobsters are Munidopsis.
I wonder why they choose the wood.
Probably delicious because they're chewing the wood.
Oh.
There's a lot of A lot of organic matter here. The bacteria will break down the wood, and the squat lobsters are here for snacks.
The wood's a feast compared to what else is around there.
Nice.
Why wouldn't you live on wood?
Good one.
It's enough for us and our children and our children's children.
Yeah, pretty much. Oh, there's a little hymenaster on there. This is a slime star.
Oh, nice.
Or is that pteraster?
Two of them.
Might be pteraster, actually.
Is that what looked like a gnocchi? Oh, what was it? A ravioli?
A gnocchi.
We don't want to collect this.
Yeah, I know.
It's gross.
Okay.
They're called slime stars for a reason.
Oh, do they have a lot of mucus?
Yeah,
I-
they make buckets and buckets of mucus.
I know.
I think May would prefer if we didn't have mucus in the lab.
Oh yeah, get a little closer.
Yeah.
But they do kind of look like raviolis.
Yeah, they do.
Yeah, I'm trying to get a little closer here. Yeah, that's...
Yeah, this is a really cool one, though.
Yeah, all sorts of critters in there.
Well, maybe Chris Mall will chime in about it later.
All right.
He's a sea star expert at the Smithsonian.
Zoom out.
Yeah, come on.
Well, that was a cool find.
Yeah, big stick.
And is the stick there a result of the steepness of this slope, and it just is falling and rolling down out of the forest and on the island, floating out, becoming waterlogged, and then just rolling down the slope?
Probably. It's a good guess as to what happened. We'll never know for sure.
Yeah, I mean, this drifted from far away. The closest point of land is over 70 nautical miles here.
70 nautical miles away from the land.
Yeah, that's the closest point of land here.
Coming up, Sarah.
Yep.
And that's due east, the current, yeah.
And we're now at 1,890 meters.
A question about the slime star.
Yeah.
Do those come in different colors?
Oh yeah, they can be like purple or pink or white.
Megan, how far are we from waypoint 5?
We are 400 meters.
Roger that, thanks.
What about that rock, Mang? That top edge on the left.
I think these are going to be similarly weathered and a little too crumbly for
Crumbly.
us.
Yeah,
Yeah.
they look
Yeah.
really altered.
So unfortunately, I think we lost our good rock zone, but
What are these little?
There's some zoanthids on that rock, and I saw a bryozoan. A couple different kinds of chrysogorgia.
Push in there.
Yeah, these are zoanthids that we're zooming in on.
Is that single-celled animal you talked about earlier, Megan, that creates that structure around it?
No, these are actiniarians, so like related, they're corals basically that we're looking at right here.
Oh, I see the structure. I see the shape, right.
Yeah. You might see these in your aquarium. There's a lot of zoanthids that make good aquarium
come on.
fauna. There's a nice bamboo coral. And a sea cucumber. There's an anthomastus, a mushroom coral. Pretty yellow crinoid.
Crazy.
There's a whole bunch of stuff. I see 6 animals.
Shrimp.
Yeah, there's
Yellow
2
crinoid, and
shrimps, a
that's,
crinoid, there was a sea cucumber, and an iridogorgia.
Iridogorgia. Yeah.
Oh, there's some brittle stars.
You push in there? Yeah.
And it's a little sponge.
Okay. Come on. Up the ridge we go.
Yep, let's go on an adventure.
Yeah.
Do we know how steep is this incline?
Yeah, we looked at it last night, so I mean it was 2.5 kilometers, and the steepest slopes were about 30 degrees, so it fluctuated between about 15 to 30 degrees. Yeah, and this is kind of right now the part we're going at between waypoint 4 and 5 was the steeper part of this ascent. Once we get to waypoint 5, it'll start flattening out a little bit.
Bridge nav. Yeah, Yeah, can we make a 30-meter move 320? Thank you.
Another sea cucumber.
Just a little bit of a time check here and looking ahead, especially for our scientists ashore. So we currently have 2:30 here, just about 2:30 past local time. Today's dive is we'll need to be back on deck by 8 o'clock, so we'll be coming off the seafloor about an hour prior to that. So that should give us about 4.5 hours of bottom time exploring this until yesterday unmapped seamount. We'll hope to get close to the summit by the end, so we still have a ways to go. A little over a kilometer or so to get to the final waypoint, and that was not quite at the summit, but yeah, it's close to the plateau where it starts flattening out. And then after securing the ROVs on deck tonight at 8 o'clock, we'll transit further to the southwest to our next exploration target, an unmapped feature or partially mapped feature that we'll map overnight and then come up with another plan and launch ROVs tomorrow morning right after breakfast. And for those folks who are on our scientists ashore that are in the continental U.S., just a heads up that in many states observing daylight savings times and adjusting time, so there'll be a shift in the clock falling back. That time change does, Palau does not change your clock this time, so just keep that in mind that the times in the dive plan that is being sent out is going to be off by an hour for those places that are falling in a place where there's a change in time.
Bridge, Nav. Yeah, can we move 30 meters 320? Fantastic.
So Daniel, we have a question about deep sea exploration. You mentioned that before they
Let's go
would
look at
use
that.
nets to scoop out specimens. How long ago was that, and when did ROVs start coming into the picture?
Yeah, great question. Maybe I'll just get a quick zoom in here,
Yep.
and then I'll
Go for a zoom.
Oh.
Tip of your tongue.
I can't remember what it's called. I know this one.
Me neither. I know it too.
It's got the cool pedicelaria around the outside. Pedicelaria are like the little projection-y thingies that aren't tube feet. So it's not a cookie store. It's a, oh man, why can't I remember?
Is it a valvatita or velatita or what?
Mediaster?
I guess.
Goniaster?
I think Mediaster, is that right?
Mediaster?
Yeah, I know.
Well, we can move on.
Okay.
And
Yeah.
and then I'll
Got a good picture.
go look at the pictures.
Yep.
And I'm going to tell you what I think it is.
Stumped Megan.
Yeah.
Yeah.
That's a rare.
That is a rare occasion.
Yeah.
I think it looks like a mediaster.
If it stumps Megan, it probably is worth collecting.
Oh, I think we've collected one of those before. Well, the Okeanos collected one.
Yeah, I was just looking at it. A couple of photos look similar.
I'm starting to think if it's unknown to Megan, it's unknown to science.
Sometimes.
Well, yeah, I'll just point out here, yeah, so Megan and the folks in her lab there at the Deep Animal Research Center and previously the Hawaii Underwater Research Laboratory have been really integral to pulling together data from various different streams, various different platforms that have been doing deep sea exploration in the Pacific and other places. And their lab has been heavily involved in annotating those videos, and a big part of that effort when they start off is creating a library to taxonomic key of what is what. So I've been creating these wonderful species ID guides.
It's low faster. That's my ID, low faster. See, that's what's so great about the animal guide is, it's hard to hold all those different animal names in your head. And we get updates to taxonomy all the time. Using the animal guide, we can use it as a reference as we're doing our annotations in order to best inform our annotations. When I throw things out here during the dive, I'm pretty confident. Oh, look at that!
What?
Fish!
Oh.
Sorry. I can be pretty confident in the IDs, but it's always good to consult the guide and consult an expert, because sometimes things that look the same can be different. What is this fish? It's looking very
Push in
Chonocops.
there. It is looking.
Like a tiny baby. Oh, it's so cute.
That's the first translucent sort of fish I've seen so far. What
Jeff?
is it, do you think, Megan?
So this is a type of anglerfish. So in the group, the anglerfishes. I wonder if Jeff's in the lounge, if he wants to catch this.
This will be the first time I've seen an anglerfish. Sorry.
Jeff is online.
Yeah, you notice that
Science
the gill openings are actually behind the little pectoral fins, the legs, if you will.
Right.
So I think this
And
might
the-
be a Chaunacops.
And the fins almost look like rudimentary legs.
Okay, Jeff.
That's what they use them for.
Jeff is interested in a collection,
Yeah, so I was thinking.
Okay.
and he
We're
says
gonna
that it's a solocisquama.
Is it small enough for a slurp?
Okay.
It swims like pelicans fly. They don't flap
Can you
their
come up on
wings
the
much.
winch a little bit?
Yep.
Under.
They can actually move water out of their gill openings. They can open and close them and use them kind of as jets.
All right, what are we thinking for a collection here?
We're gonna slurp it.
Yeah,
You're going to have to get ahead of him with the slurper.
Come wide on the
Yep.
camera there.
He's drifting with the current, no doubt.
They're not good swimmers, so you should be able to slurp it quite easily.
So down on the camera.
Tilt.
He's moving. So I wonder when we talk to Jeff whether this fish, if he's related to a anglerfish, has any bioluminescence ability.
Which
These ones don't.
canister would we like?
so this can go in number 3.
You said it was an anglerfish, right, Megan?
Yeah, yeah. Sometimes they call them like coffin fishes or frog fishes.
So this type of anglerfish, Megan, does not have any bioluminescent ability?
No, these ones that sit on the bottom do not. What did Jeff say this one was?
He said it's a solocisquama,
Solocisquama.
and it's in the family Ogcocephalidae.
Okay. It's gonna fit in there. I don't know.
Yeah, it looks a little bit wide.
All right, we're up
You're
on
nicely
3.
positioned.
All right. Start suction.
That's it.
Suctioning.
It's like catching a fly ball out in left field.
Ooh.
Oh, there he went.
Oh, up.
Poor Jake is like hyper focused, and we're all.
He's not quite, that
I'm,
fish, is-
I'm laughing too. It's
Okay.
funny. Yeah.
that fish is not as helpless as we thought.
Okay. Jeff says you could get the net. Oh,
Oh, it's in there.
It's in there. Okay. That moved pretty quickly.
Oh,
There
there
it
it
is. All right. We're good to
That's
go.
fantastic.
Okay. That was very quick.
Oh, that's awesome.
It was a perfect fit.
A little dizzy.
My day is made.
Oh,
Yours is made.
Great job. Fantastic.
all right.
There you go, Sarah. your
Nicely
first
done.
anglerfish.
56.
56.
Yep.
That was awesome.
I think that was the highlight of our watch.
I think Jake can catch anything.
Jake's a fantastic pilot.
Oh, thank you.
I agree.
I'll second that.
Quite fun to
I would
film
third
with you.
that. Very, very calm.
Make
Oh,
a
I
great
just
he'd
want
make
to
it-
keep looking at it.
He'd make a great shortstop.
Well, we don't want it to swim out. It might get
Let's
ideas.
Move
From the urchin.
clockwise.
Excellent. As we move on here, maybe I'll just get quickly back to Imada's question there about the history of deep sea exploration. You know, what was considered really the first deep sea expedition that was done by the Challenger in the 1870s, so 250 years ago. Our first world voyage just for scientific purposes that went all around the world. That expedition collected a whole bunch of samples with deep sea dredges and trawls. It also sounded a whole bunch of places in the ocean. They actually collected a sounding close to the deepest part of the ocean in the Mariana Trench. So they basically send a line down with a weight and then see once it touches bottom and then pull it back up. And they recorded a sound of over 8,000 meters back then 250 years ago. They also...
Who funded and organized that trip? That expedition, excuse me.
That's a great question. I believe that's
the British.
Yeah, it is the British. Yeah. Do you know a little bit more about it, John?
I've heard of it. I just know I think it was done at the height of the Victorian era, wasn't it?
Yeah, I believe so. So yeah, I went around the world and it's very interesting. I mean, still a lot of, when I did my graduate work, I described a couple species from samples that were collected back then 250 years ago and had been sitting on shelves and then people hadn't looked at them for a long time. So yeah, they collected a whole bunch of dredges and trawls and sampled and that really changed people's perception. Back before then, people thought about the deep sea as a lifeless emptiness, and that expedition brought back all kinds of interesting looking creatures that people had never seen before and changed that perception of, before then you had the, people referred as the deep sea as the lifeless part of the ocean, and that changed that perception.
Push in there.
And then actually there's early submersibles in the 1500s. Just submersibles, non-tethered, obviously not deep diving submersibles, but people going down in a chamber. I believe the first ROV is in the 1950s, is a Russian ROV, the Poodle, I believe that was. And then in the 60s, the US Navy started working with ROVs, and obviously rapidly changing technology and adapting technology. So that's, prior to that, a lot of human-occupied vehicles. Obviously, when you put a human down to these depths, there's a risk, and now technology is so good with, we're able to basically experience almost the same thing people are seeing in a submersible. So, and we're starting to see a new transition too in terms of into the autonomous vehicle, so that you don't have a human steering it, but you basically send a bunch of commands to the vehicle, and just the vehicle goes and does its thing. So there's a little bit of an evolution. Every technology has its advantages and its trade-offs. I'm a firm believer in this telepresence-based model.
Absolutely.
It's an absolutely wonderful way of doing science, and so hopefully that addressed the question by our curious viewer there. Thanks so much for all those questions and keeping engaged and
those earliest submersibles that you mentioned, Daniel, from the 1500s, my memory was that they were created to go recover Spanish treasure, treasure from treasure ships, which would make sense. They were going after trying to recover gold or silver. A little hard on the divers in those days. It wasn't very safe.
Yeah, that, and obviously also in warfare, as always, a way to get underneath the ships of the enemy.
Similar one.
Maybe this one will be more cooperative.
Go for it. Zooms away.
This is a cusk eel. Probably the Bazazetus that we keep seeing, these shallower ones.
Nice. All right. Come back wide.
That one was more cooperative.
May? Oh,
Yes?
we have 2 questions about that fish we just collected.
Yes.
First question is, can you spell out the name of the fish?
Of course, it's SOLO, solo, CISQUAMA, and the family name is Ogcocephalidae. And you can thank our lead scientist, Jeff, for sharing some of these accurate spellings. Even he misspelled it the first time and had to correct himself, so.
Okay, and then the second question, will the pressure difference from here to the surface hurt the fish?
Yeah, well, definitely all of the specimens that we collect, we try to collect as little as we need in order to document and understand the environment. But
Push in there.
these specimens sometimes
it's
are
a little
alive
baby
on
eel.
recovery
Baby eel.
and sometimes not. And we try to process them quite quickly, so they would be sacrificed to science, but as I said, we really try to collect as little as possible to characterize, and often we opt to image and get good video. So most of the time, all of these animals we're looking at, we're getting good imagery about their behavior and morphology, and then just occasionally doing a collection.
Yeah, that was a juvenile synapobranchid eel, cutthroat eel.
So Jeff asked if we could check out every small fish that we see. Or like very small fish.
Okay.
I think that was the one that we just saw. Was it? Yeah.
Bridge, Nav.
On that fish, their common names are the bat fishes.
Batfish. Can we move 30 meters, 320?
On one of my first Nautilus expeditions in 2015, about 10 years ago, we found a pancake batfish, and it was a new species. I always thought that common name was really cute.
Yeah.
Pancake batfish.
Oh, another fish up here.
Makes me hungry.
Push in there.
There's a little one right there.
Seems to be a lot of these ones.
Yeah.
Nice.
Well, it may not be that little. Is it longer than 20 centimeters?
Well, remember the lasers are in a lot further away, and
Yeah.
we're
Oh,
zooming in on it,
That's
so
right.
this is actually really
That's
small.
Right. That's right.
Oh, yeah. I don't even think I can get the lasers on it. It's too.
Yeah, and I think it's too small to get the lasers on it.
The laser itself is bigger than the width of its end of its tail.
Yeah.
Yeah.
All right.
One quick comment on that earlier question. So, in these depths and in the deep sea, many deep sea fishes do not have swim bladders, and it's an organ that many fishes that you find in shallow waters have. Now, for those fishes that have swim bladders, a rapid ascent would cause a lot of harm. I actually had the pleasure of diving with a lot of deep sea aquarium fish ichthyologists over the year, and when they catch fish for the aquarium trade at relatively shallow depths for considering what we're doing here, but when you dive in 100, 200 meters of water and you bring up fishes from those
This
depths,
little tiny fish at the bottom.
when you go up, you have to decompress them. So you do a little puncture in the swim bladder and,
Yeah, the
and
smaller
basically
one.
let the gas vent so you can keep them alive in the aquarium afterwards.
All right, you can push in.
I can't remember
At the bottom.
if I have a vague memory of some scientists wanting to try to create a chamber that would maintain pressure to collect fish at depth, but I don't think it was successful. Have you heard of something like that to avoid that decompression?
Yeah, I've heard of it. Yeah, I haven't tracked it.
Yeah.
I mean, it's a massive endeavor.
Yeah, certainly.
I think Jeff has a chamber like that. You can bring it up, but then what do you do?
Yeah, that's the thing. Then you have to maintain that pressure, or if you want to keep it alive, or.
I believe they have a facility like that at Southampton in England. Oh, that's a nice Basacetus. They've been able to keep vent shrimp alive for long periods of time.
Yeah, and in those cases, it's typically to do studies to understand their physiology, what their respiration rates are, and those types of things.
Oh yeah.
That's
Yep.
a nice
Yeah,
one.
let's
That's a
take
good one.
look.
That is a large fish.
It's probably Coryphaenoides.
Push in.
So this is a rat-tailed fish or grenadier.
Unlike the one we saw yesterday, this one looks very healthy.
Mm-hmm. These guys are the scavengers, top predators down here.
I'm going to say he's big enough, as we say, a big bass in Texas. He can eat anything he wants.
Yeah, but they don't taste very good. I didn't eat it, but some of the grad students decided to eat one of these. Said it wasn't very good.
I'll stick with bass.
Yeah, I think bass is a good choice.
I'm not sure who's monitoring the ROS proc screen, but there's a little low disk,
Oh, that's great.
disk space comment.
There goes the NDIs.
I don't know if is that an ROV? The ROS proc is got a low disk space warning.
Okay, that's a.
I don't know who's.
Yeah, either Chris or Mike can deal with that.
All right.
I'll send it now. Thanks.
Yep. Want to see if the NDI is still up. Remember that thing Dan showed us? And then I think it's Studio Monitor.
Daniel, are you notifying them? Yeah. Okay. Thanks.
And then top left corner, you should be able to see all the streams that are hit that one right there. Yeah. Stereo. No, it doesn't look like they're up. So maybe they got power cycled or something. I don't know. You can close that. I'll just go back to normal dive salvo here for a second. All right, come wide on the. Yep.
bridge, Nav.
Looks like they turned
We
up.
move 30 meters, 320. Thanks.
We are now at 1,823 meters.
Got here, is that a shrimp or a fish? Shrimp. Yeah. Ooh. There's a fish.
Question for Megan.
Okay.
Pushing
When you're
on this
asking
fish.
the ship to move 30 meters, how fast does it move?
The ship is moving at 0.3 knots.
Okay, thank you.
So very slow.
Come on.
There's another of those fish.
Nice.
One more Coryphonoides, please. Jeff's probably having a great day right now. Just keep seeing a lot of fish.
Push in there.
It's another big one, too.
Mm-hmm.
Who also looks pretty healthy.
Nice. Alright, come wide. Let me see if I can. There's another fish
Yeah,
right
it's
off
got
the
a
screen.
few parasites on it.
Oh, yeah? Where'd the other one go? Oh.
Yeah, there's the other one.
Jeff wants all the fish.
Yeah.
Pushing there.
Jeff really likes fish.
These ones are tough. They're
Yeah, they're fast.
tough to get a zoom on.
Jeff is very pleased. He says this dive is great in all caps.
Alright. Come back on.
There is also oh, there's a Cerianthid in the sediment right there.
Oh,
See the red.
pushing there.
Something was living in there? Oh, no, I see.
Yeah, so this is a tube anemone. So you can see it has a tube that it lives in. You can tell this
Okay.
from other anemones because there's an inner ring of tentacles around the mouth. It's a little hard to see from this angle.
So when it detects a fish come by, it emerges from that tube and grabs it. Can it propel itself out of the tube?
Oh, that's not how these guys work. They're filter feeders. So if a fish came by, it would shook back into the tube.
He just makes the tube to anchor himself.
Yeah, it's for protection. Looks like we got a lot of sediment. Made it sort of to the top of this ridge. So there's probably going to be even more fish. Bridge nav.
Oh, so the fish.
Can we make a 40-meter move, 320? Thanks. Oh, there's another fish. Looks like an eel.
Push in there.
We have some interesting facts from our scientist ashore, James Raymer, who says that the tube is actually a special type of nematocyst.
Oh, neat.
And it seems actually that some of those cereanthids can eat fish, maybe larvae.
Oh, do they? Oh, fish larvae.
May, what does that mean, a pneumaticist?
It's like a cell
Yeah, it's a
in
specialized.
in jellyfish, right?
Yeah, it's a specialized type
Right.
of cell that are found in a lot of corals and jellyfish, and a stinging cell basically.
Yeah.
And
Venomous.
So this is eel in the subfamily Iliophinae, like the Iliophis, an arrow-tooth eel.
All right, come on.
yeah, quick shout out to our data engineer. Had a little, as there's always something to fix, but yeah, we were working with a Norbin and the GUI, and so thanks so much, Mike, for fixing that.
Way to go, Mike. Keeping the show going.
Yeah, it's the first time I've been like paying close attention to that interface. It is spectacular. That's very helpful. I'm gonna have that on every dive here.
Oh, the Norbit's on?
Yeah, when we can. Yeah, it's really, really, really helpful.
It's on PC4 if you're looking for it, Jake.
Okay. I think it might be here too. Oh, no.
We have a question about the
Oh, I was
arrow-toothed
on my
eel.
left. That's why.
Do we know what is their depth range?
They live pretty deep, you know. We'll see them at these depths and deeper, for sure. So the arrowtooth eels are in the same family as the cutthroat eels, so the Synaphobranchidae, and we'll see them shallow on seamounts all the way down to abyssal depths.
Yeah, a couple fun facts here now that John Spack, and we talked about the Challenger expedition. So in the 1872 to 76, so 4 years around the world, funded by the British government as the first really systematic exploration to go around the world and explore the deep sea. So 4 years, when it was all said and done, they had done over 500 deep sea soundings, over 300 trawls and dredges to deep sea habitats, and the specimens that were collected from those 4 years of explorations led to the description of close to 5,000 new species at the time. So it was a massive, massive scientific advance at that time. The findings from that expedition continued to be published many years after the expedition was done. I believe it was close to 20 years that they continued looking into the data.
Extraordinary.
Various different volumes, close to 30,000 pages of scientific exploration. Now, when you look at the amount of data, and if you scan all the data that was collected in those 4 years, it could fit on a small thumb drive that we have today. And on our expeditions, we collect many, many more times because of the resolution and the data that we collect. So yeah, it's a very skewed in terms of how much information we are now able to collect. And yeah, just wanted to use that opportunity to also thank our unsung heroes, our data engineers that work tirelessly behind the scenes.
Absolutely.
It's just it's a very hard work and really, really so integral to the discovery process.
This is a Bolosoma, another type of glass sponge, also in the family Euplectellidae. And then there's a little Ophiocanthid brittle star underneath. There's another kind of brittle star on a cobble up at the top of the screen as well. So different kinds of brittle stars all over.
Nice.
That's great, thank you.
All right.
The stocked sponge that we collected yesterday is really interesting to see. We have it, part of it preserved in ethanol and the other part drying, and you can really clearly see the stalk being made of these silicate woven spicules. It's really neat. I've never seen something like that collected or up close, but you can tell how strong they are, and that the organism actually kind of weaves its stalk with these glassy, stick-like spicules.
Yeah, that was a really cool collection.
Yeah, definitely. If anyone wants to look who's on the ship, come to the lab. It's definitely worth taking a look.
So a viewer wants to know what is the difference between this mapping that the ROV is doing versus the mapping that was done prior to the dive?
Yeah, really a great question and very important one. So in essence, like in terms of the fundamental technology that used, that there's both sonars, so it's basically a device that is mounted. What we use to map this site is a multi-beam, so sonar that's mounted on the hull of the ship, and we basically then transmit a signal down to the bottom of the ocean. It bounces back, and then It gives us information about the depth and texture and of the seafloor. You can put that same kind of device on a remotely operated vehicle, and then you get closer to the ground, basically. So in essence, the technology is the same, but when you get closer to the seafloor, as we are with the ROV, we're now just a couple meters above, so the resolution is much higher. So the resolution.
Excuse me, I'm sorry. Is that a skate egg on the left of that rock, that big rock? I'm sorry to interrupt you, Daniel.
No, they're great, yeah.
What is that? See that thing leaning up against the bottom of the rock?
Dead sponge?
It's
Is that just a piece of wood?
I think it's a piece of wood.
Yeah.
Okay, I'm sorry to interrupt, Daniel.
No, please do. There's always things that we don't spot. Thanks, John. Yeah, let's take a quick.
Can you come up on the maybe 5 meters? I just see that tether down there.
It looked flat and brown and about the same. It doesn't have the points
it
on
is
the
dead
end, but
sponge.
it looked dead sponge.
Dead
Yeah.
sponge.
Forgive me, Daniel. Please
Oh,
continue.
Oh, no. I appreciate that. Yeah, there's always the unexpected that might be there. But so, when we have a sonar on the ship, there's several thousands of meters between them. And so the resolution of what you can map has to do with several things, but the distance to the target will kind of dictate how fine of a resolution you get. I mean, you can get a little bit higher by going slower and pinging faster. But at these steps, we typically get resolve when we map with a ship, things about 50 meters or so, sometimes a little finer. Now, because the multi-beam on the ROV is just right above the seafloor, you can get down to sub-centimeter scale, so you can get really fine. I mean, in fact, some of those maps that you can create with the multi-beam mounted on the ROV look, very similar to photographs because it's so high resolution. You don't have the color, but yeah, you just see the fine details. But now the one issue is, when you map with an ROV, you're just mapping directly what's underneath the ROV, so you don't really have much of a swat width. Versus when you separate the distance with the sonar that's on the ship, you're basically have a swat width. It's typically about 5.5 times the water depth. So at these depths, about 2,000 meters, you can map 10 meters or 10 kilometers across. So yeah.
Daniel, so this side scan orbit sonar that's on the Hercules and is operating right now, as Chris, one of our expert data management video guys, experts who put this on, you'll be able to see a shell casing, apparently. You'd be able to spot another one of those 75 millimeter shell casings.
Yeah, you could,
Yep.
Absolutely.
We've used it on Ocean Networks Canada to find small instruments on the seafloor and about the size of those casings and stuff, and you see them light up in the sonar and clear as day, and you can return to that spot. It's pretty amazing.
The detail is extraordinary, and for any of the listeners that are interested, if they'll go to the expedition that was done last October off of Hawaii, where the last one I did one I participated in last October. I scanned a Japanese submarine that was sunk off the coast of Oahu in 1945, one that was captured for the Japanese by the American Navy, and the Japanese sub was capable of going up to 50 knots underwater. It was 500 feet long, and the side scan sonar showed it in incredible detail. It was as though you were looking at a photograph of it. It's a remarkable instrument, and if you're interested, folks, you can go to the website nautiluslive.org and look at last October's expedition off of Hawaii and see some incredible footage of very intact Japanese submarine on the bottom of the ocean, both with a side scan sonar and with a brand new revolutionary ultra high definition 180 degree color video that was taken as well.
Those are amazing models. Hi, everybody. I'm guest interloping here.
Hello, Megan.
Hey, Jake.
Yeah.
Is it okay if I test with the zoom in and zoom out on the cinema cam?
Go for it.
I don't want to startle you.
Yep. Nope.
Okay, thank you. I actually don't even know where I should be looking. It's that low one, yes?
Yes.
Okay, perfect. Thank you. I'm not seeing any change there. No, that's okay. I can kneel. Oh, did it? Okay. Oh, okay, there we go. Rapid zoom.
What's that?
Oh.
Fish.
All the way in. Oh, okay, so it just has 2 stops.
Looks like it's sitting on the bottom.
Yeah.
Okay.
Push in there.
It's a tripod fish.
Yeah.
Bathipteroi. I can never say this one right. Bathipteroi atricolor.
These are so cool.
I love their scales. I want this pattern like in my bathroom. I feel
Ooh.
like, wouldn't that be nice?
It'd be a really nice tile.
Megan, do the tripods at the bottom of the fish serve as feelers for the bottom so we can tell where the bottom is? Do they have any structural purpose, or what function do they serve?
So they're ambush predators, and so they'll sit and perch on the bottom like that on the tips of their fins, and they have these really nice like filaments coming out from the head. You see those? And that's how they're kind of
Yes.
detecting movement in the water for their prey.
I see.
They're
And
on the
pitch, it allows them to detect their prey in pitch darkness.
Mm-hmm, exactly.
And they're isolating their own movement by being up on stilts.
Exactly.
All right, come on.
So a follow-up question about the ROV mapping. Does it also have capabilities to do photogrammetry?
I mean, you could. I mean, photogrammetry is basically just a technique where you take periodic photographs and then you go on a pattern along an object, and then you stitch all those photographs together to create a total model of what you're looking at. So you absolutely you could do something very similar.
Or pair them together.
Yeah.
The 2 approaches.
We're going to do a little pilot swap here for the last 30 minutes.
Let's go, Sarah.
Yeah, you gotta pull that seat forward.
Okay, so you're in auto heading. I left you in auto heading.
Alright.
Yep. Kind of maintaining an altitude of like 2 to 3 meters and just moving along pretty slowly on the bottom. So you've floated up a bit.
Mm-hmm. Coming down.
Yeah. And then you're just directly under Atalanta, so you got some room to fly ahead.
Okay.
I think I had your joy gain at about 50%, which is nice for flying nice and slow. So.
Okay. Gotta get a feel for it.
Yeah.
Been a year and a half.
So a question to our pilots, if that's okay.
Yeah, sure.
Is there a difference between driving Herc and Atalanta?
Yeah, so it's different different controls. In Atalanta, you're really controlling the depth and the heading. So you're paying out the winch. Atalanta's connected to the ship with the 6/8 cable, paying out and hauling in the winch to control the depth. And then the heading to keep Hercules in view. And then with Hercules, you basically have 6 degrees of freedom. You can move up, down, left, right. You can change your heading. It's a free-flying vehicle. So it's a bit different than Atalanta, where Hercules, you're kind of flying it. Atalanta is a little bit different.
Thank you.
I have to get an idea of like the depth.
Yeah, so I like to look at actually the upper cinema cam. You can see there's like a shadow from the porch.
Yeah.
And you can, as you get closer to the bottom, like if you were to come down now,
Disappears.
it kind of shrinks, that
Yeah.
shadow. So I use that as like a depth
That's a
or
good one.
altitude kind of proxy. And then I look, there's also altitude there on the bottom.
And there's
screen there.
altitude
Yeah,
here,
I
and there's
keep
altitude
looking.
there.
That's where I've
yeah.
been
It's in like
keeping an
8
eye.
different places if you look for it.
I
That way you can see it no matter which screen you're looking at.
Yes.
It's helpful.
I was used to gauging it. Usually we'll have like a little bit of the porch on the bottom, and so I've always gauged it with the shadow there, so these are just different.
And if you want to get a feel for it, like you can turn the joy gain all the way up and kind of get a feel for, I don't know, how the vehicle moves with full beans.
I found if I do that, I like to speed.
It's very tempting when you get excited about something. I do
you,
it all the time.
You could like speed up just a bit though.
Okay.
You're kind of falling behind Atalanta.
I'm usually stretched out, which can cause me to get, a little too far ahead, but I like to be a little stretched out, especially when you're climbing any cliffs or surfaces, or you're moving along at a decent pace. It's good to be ahead, because then you can look at things and spend some time. I'm coming up a little bit.
Yeah. Tether starts bumping up behind it.
Yeah.
Yeah, he fell off the screen, so it's like, where'd you go?
Yeah. I know I'm constantly adjusting the camera over there because it's behind, it's under, it's in front.
Yep.
Yeah.
Always trying to keep it centered.
Megan, do you have a sense of distance to waypoint 5?
Oh, we're really close.
Wonderful.
We've got like 100 meters to go.
Excellent.
We're at 1776 meters.
So a question for our science team. Do creatures from in the deep ocean behave any differently between daytime and nighttime, despite it being dark all the time?
Yeah, that's a really good question. I mean, at these depths, so like obviously the ocean is stratified in many ways. In about 1,000 meters and shallower, which some people still would consider that pretty deep. In those you have vertical migration, so you have a lot of species that on a daily basis they will move up into the shallows at night and then go back down when it's daytime. So that's not limited to some organisms that live at 1,000 meters and shallower. In the deep sea, where below that 1,000 meters, you don't have that anymore. And yeah, to the best of my knowledge, but yeah, Megan might know more. Like you don't have those kind of circadian cycles of sleeping and waking and moving at certain times.
What Daniel's just described is something that Professor Jeff Drazen told me the other day that is extraordinary, that the largest migration on Earth is the movement Daniel just described of these creatures from about 1,000 meters down. When the sun goes down, they go up because to feed, and they do so to avoid predators, and then when the sun comes up or the moon comes up, they'll go back down into the darkness to avoid being eaten. And it is the largest migration of animals on the face of the earth in terms of mass, in terms of numbers, and it's also extraordinary. Dr. Drazen of University of Hawaii pointed out to me that these animals are so small that their movement of hundreds of meters compared to their body size is absolutely extraordinary, and they're doing it twice a day. It's extraordinary what scientists like all of you have taught us about the oceans and their importance to the earth.
Oh. Caught a little sediment there.
Happens.
It's definitely
A follow-up question. Would the moon phase affect their reproductive behavior?
Excuse me?
I was just going to actually bring that up. Yeah, it's a really...
Could you repeat the question? I'm sorry.
Would the moon phase affect their reproductive behavior?
Yeah, the migration behavior, absolutely. So I mean, and they've measured that these animals that undergo diel vertical migrations, they follow isolooms. So those are places where you have a certain light level, and so they're basically moving up to that light level. And so if there's a full moon, that will affect when the timing and how much they migrate. Yeah, and I mean the whole history of how this was discovered, and there's also very interesting theory behind it. Now, people had known about these vertical migrations for a really long time. That's been recorded, and there was speculations of why that might be the case. Now, there's an energetic benefit from that. You can go up in the shallows and eat, and then go back down and digest in a place where it's cooler, and there's like metabolic advantage of that. So that's one theory, but the predator avoidance theory really won over the years, is that because they saw that actually some organisms do the reverse pattern, and yeah, the most telling piece of evidence is that the lunar cycle, or where light is an eclipse and things like that, they've measured basically that the migration pattern was disrupted. So these organisms have ways to detect light, and then they follow a very strict light level as they move up and down the water column.
So you want to frame it up like just below the lasers, and it might be hard because you're in auto heading. So you come out of auto heading, you'll have more heading control.
Dan's trying to talk to you.
Oh, really?
Yeah. Hold on 1 second, Dan.
What's up, Dan? You in the data lab? Yeah.
Come up there.
I'll do it. Yeah, we're fine with that. Yeah.
Another interesting kind of astronomical force on deep ocean ecosystems actually occurs in hydrothermal settings as well. There can be effects on the
Closer
the
to-
flux of fluids coming out of the seafloor from the tidal forcing, even at great
So little.
depths. And that was something that
tiny.
that was a project I was involved with earlier this year. Some researchers interested in this tidal forcing and how that can impact the hydrothermal communities. So very different ecosystem than what we're looking at here, completely different. But it is really interesting how the sun and astronomical and gravity forces can impact
I think that's good.
environments from
Alright. Zoom out.
the surface to the deep ocean.
May, you're saying that the tidal forces from the moon can be detected by these animals as deep as the hydrothermal vents and affect their movement in the deepest part of the ocean.
Yeah, and at hydrothermal vents, the animals there, their source of energy is coming from the rich, chemical-rich fluids coming right out of the crust through a process known as chemosynthesis instead of photosynthesis. And that flux of fluids coming out of the crust can be impacted by tidal forcing. So can actually impact the communities around those areas, even in complete darkness, and even in areas where we wouldn't expect a very strong tidal difference. You know, when you're at sea, you can't really notice a difference in tide necessarily as you can when you're in a shore-based environment where you can see really clearly that a tidal change might be pretty significant. But even out here, we have differences in tides, and they're felt in those communities and in hydrothermal systems as well.
As a lifelong amateur astronomer and an absolute passionate follower of oceanography and space exploration, I learned that the moon, when the moon was formed, it formed much closer to the Earth as a result of an impact. We know from the Apollo missions, a Mars-sized planet hit the Earth about 100 million years after the Earth's formation and blew out this big blob of material from the Earth that created the moon. And of course, it was tremendously hot, but the moon formed very close to the Earth and gradually moved outward. And the effect of a moon that close was to churn the oceans. When the oceans first formed, the gravitational pull of the moon was so huge that it was like putting the oceans in a giant blender. So there was, talk about tides. I mean, I can't even imagine how huge they were in the early phases of the Earth's history, the early phases of life. The churning motion of the moon, which is much closer, that whipping and churning of the tidal forces was apparently critical to the formation of life, creation of life on Earth.
Yeah, it just shows you how interconnected habitats and environments and planetary connections are from the really fine scale to those big planetary scales.
And how essential the oceans and water is to the creation of life, which makes the mission to Europa all the more interesting and exciting, because it's a worldwide ocean that looks like Earth's is as old as the 4.6 billion years. That ocean on Europa is as old as the one on Earth, and has all the same chemical elements. And it's very exciting to think about what we are learning here about the ocean on Earth has application throughout the solar system and the universe.
What's that little guy?
Sea cucumber.
Cucumber?
Oh, there's a little jelly right above the lasers.
Oh yeah, there's a challenge for you.
Oh, nice.
Chase that jelly.
Probably get a quick push right now.
Yeah, can we push it on that? Get a little closer here without disturbing it too much.
Nice.
So on that vertical migration, we have a viewer that's been listening and notes that smaller organisms participate in that type of migration, but are there any bigger fish or other animals that migrate?
That make a similar migration?
Yeah.
Yeah, I mean, I believe there's, yeah, at all scales, from tiny copepods, things that are microscopic that you can't see, to like bigger fish that participate in this. And so the reason really is that there's more food closer to the surface. And so you want to get closer to the surface where there's more food, but there's also more predators closer to the surface. So to balance those two forces, you go for those things that are visual predators. You go when you can mask yourself when it's nighttime, and you can go up and feed and not be detected. And as I was alluding to, there's actually some species too where their predators migrate, they will have the inverse relationship, so they basically are moving away. And yeah, it was very interesting in terms, as I mentioned, this had been detected long time ago. You can actually see it in mapping data. You'll see what's called the deep scattering layer, that this is massive in the water, that's scattering the mapping data, the sonar data. So there's a lot of biomass, so life that is the mapping data, the mapping sonars is bouncing off. And so this has been known for a really long time.
Dr. Brian Kennedy told us the other day that he witnessed one of these migrations of some of the very small animals and some of the larger animals, as Daniel was just saying, in a canyon, an underwater canyon that narrowed down to a fairly, narrow point at the bottom. And as the migrating animals went down and they got jammed into that narrow crevasse, the predators came in and just had a field day, and because the food was all concentrated. So yes, it's a great question from the listener, and they indeed, there's when the migrations happen and there are little larger animals, even the smaller ones, it provides an eating opportunity for predators.
Back up, catch it.
Yeah, and two more things. So now this is also one of the many reasons why we're interested in seamounts, is particularly those that have a shallower summit. Those that have a summit of 1,000 meters or shallower, and we're going to be exploring two of those on this very expedition.
Click on of auto heading, it might help you.
In those cases, the migrating animals will basically come to the summit, so you'd have a extra concentration of highly mobile animals on the top. The other interesting thing about diel vertical migration, it's not just vertical, it also has a horizontal component. So in oceanic islands like Palau, it's not just that thing comes up and down, but they also will come closer to shore. So you have things that are typically found farther offshore that at night will come closer to shore.
Zoom out.
Yeah, in some previous Nautilus expeditions, working with Mesobot and Drix, which are two other vehicles that have been on Nautilus a few times, we were able to really dig into that and map these migrations with a surface vehicle, Drix, and then go follow them with Mesobot. And very similar to the eDNA work we're doing here, that vehicle was able to filter eDNA and get a sense for some of those migrating species.
Can you explain a little more about what Drix and the other vehicle are and what they're capable of?
Move 340.
Yeah, just quickly, I guess. Drix is a surface-based autonomous surface vessel, so it's used to do seafloor mapping, like what we do with the multi-beam system on Nautilus, but it can be driven by somebody remotely, or it can be pre-programmed to map. And it's equipped with different sonars, some of which are really useful for detecting those, kind of like a fish finder, but a very fancy one. And it can see those layers in the water column, and not just map the seafloor topography or bathymetry. And
Drix is sponsored, quickly, that Drix is sponsored by the University of Rhode Island and is on the back deck of the Nautilus because they're a part of the Ocean Exploration Trust.
Daniel
New
can speak to
Hampshire,
that.
Yeah, I can take that. So yeah, Drix is at University of New Hampshire. So Dr.
Hampshire, forgive me.
Yeah,
Yeah.
Dr. Larry Mayer.
Since they're bad about that.
Yeah, and so that project that May was just describing, so that was done here on Nautilus, where a new approach to studying deep vertical migration was developed here on a
Zoom in
Nautilus
on that.
expedition, and that used.
Maybe.
I'll just
Little
pause
guy
here
there.
for a second while we get the zoom in.
Try and get a little closer, little cucumber.
Is it a Hollister?
Yep, little
Yeah.
cucumber.
Nice.
It's gonna swim.
Oh.
Oh,
Have we seen this one before? It looks familiar.
yeah, we've seen a number of these ones.
Yeah.
That's always fun when they swim.
Yeah.
Nice. Is that his digestive tract, Megan?
Yes, it is. It's all full of sediment.
Try and come up a little bit.
May, forgive me, I interrupted you. You're about to tell us about the second vehicle.
Yeah, I guess just quickly. So,
Copter. There we
Mesobot
go.
is just another type of autonomous vehicle that can be programmed on a mission to go into the mesopelagic. That's why it has the name Mesobot, and it can follow and study these twilight zone migrations, and can do so very quietly and in a non-disruptive way. And
Autonomously?
Yep, and it's equipped with low light, something called a radiometer, and it's equipped with a pump system, so it can actually like filter water as it goes. So here, when we collect water for eDNA with the Pickard team, it's a collection of water that then gets filtered in the lab and can take quite a lot of time. But on Mesobot, it's equipped with a pump, so it'll be filtering in real time at a specific setting, pre-programmed. So when those vehicles are working together, it's really interesting and exciting for folks who study the twilight zone, because they can correlate this migration and then move right into it using the combination of a surface vessel and Mesobot. Maybe Daniel has more to add to that.
Yeah, I'll just add one. So the approach that was developed here, so basically you use Drix, and it has a water column EK80 mapping sonar that basically maps that deep scattering layer, so you can get very precisely see where that aggregation of life is. And then you could basically use Drix to send an acoustic command to Mesobot and go directly to that deep scattering layer, and basically direct it to turn its cameras on or off and turn its eDNA sampling systems on or off. So that's was a really revolutionary kind of new approach, because prior to that you had no way of knowing that you were actually sampling within the deep scattering layer. So the deep scattering layer also avoids things. So we actually, when you have on the ROV, when you have a sonar on and you go down, you'll see the deep scattering layer, and you'll see it moving away from the ROV. So it's basically avoiding. And so basically prior to this approach, when you were studying and trying to sample the deep scattering layer, you basically could map it, but then you would have
That's a good one.
try to deploy a net at those depths, but you didn't really know if you're sampling.
Zoom in on
And
that.
with this verified directed sample, you could position Drix directly
another
over it,
Acanthonis
see where it
armoris.
is, program Mesobot to go it, sample, and you could still see the deep scattering layer, and make sure that Mesobot was indeed collecting data in that. And so it's a really a wonderful thing that was kind of last year on an expedition on Nautilus, where we were doing an expedition, was the first time that was really perfected, and then we just used it in American Samoa, where we again had Mesobot and Drix on board. And yeah, it's just kind of the new age a little bit of autonomous vehicles, is using multiple ones of these technologies together simultaneously and collaboratively,
Struggling.
and creating some real synergy between these different vehicles.
Bagging up. Bear with me.
Okay, and then one last question on this migration. So A viewer wants to know, do large storms have any effect on the animals that migrate up and down the water column? For example, when hurricanes or
can zoom
typhoons
in.
block the sunlight or the moon.
Keep 'em in here.
So yeah,
Oh.
these animals use light as their cue to move. So once it gets darker, they'll start moving up, and you know, they've actually measured this during a solar eclipse that they will, you know,
Right.
that
we
disrupts
can let
that
this one
motion.
go.
And so yeah, you know, heavy
Nice.
clouds, heavy things that disrupt the natural
I
light
would want to
patterns
see
would
that,
have an
that pink
impact. So
thing
they're
that's up there on the
basically
sediment.
measuring the light and then following very precisely a specific isoluma or constant light level and moving up as it gets darker.
The darker it is, the safer those little animals feel.
Exactly.
Just to add to what Daniel was saying, migration is also driven by temperature for some animals that live in the sea. In the case of like tuna and
Zoom
whales,
in.
because like around here it gets pretty hot, they will come up during the early morning to feed and then go back down because the temperature in the surface gets warmer. So they go back to the deep because it's colder down there.
Try and drop down
Daniel,
a little more.
what do you
It's
think
just
of this
getting a little-
black coral?
Say it again?
What do you think of this black coral?
Yeah, I'm waiting for it to settle. But yeah,
Oh, I'm making a bit of a mess.
yeah, if it is what I think it is,
Yeah,
that new genus.
sometimes do that.
Back up a little bit.
Yeah, auto depth near the sea, right near the seafloor. If you're in auto altitude, the DVL kind of doesn't do great when it's right up against the
Okay.
seafloor.
Do you ever just land?
Yeah. Oh yeah.
Okay. Yeah, I'm struggling.
Right. So I would suggest driving ahead,
Okay.
getting out of the sediment, and coming wide on the camera.
Lose
If
it.
it's,
There we go.
yeah, just drive ahead a bit,
Okay.
and then that'll push the sediment back, and then you'll come out the other side there. I can find you on. Yeah.
To go up and like turn around potentially.
It was right underneath you.
Okay.
Yeah.
Yeah, I didn't really get a good look at it, Megan, but I think it could be that abyssopathes, but I was,
I'm not sure if
I
it
would,
was on a
yeah,
little
turn your
tiny
heading
pebble
instead
or
of backing
not.
up.
yeah.
Okay.
And
Yeah.
then maybe
Oh,
and then lateral right back. There's that fish.
yeah.
It's tough that we don't have right up the pan.
Yeah, I gotta come down a bit too.
We don't have the wide view camera, so it's hard to find it with just the
It's
zoom.
right there. It's right in the middle.
Yeah.
Oh. Right in front, right in the lasers?
Yeah, right next to the lasers.
Okay.
Yeah. So I'm just trying to slowly come forward and come down.
Ship stopped, Megan?
The ship has stopped, yes.
Cool, all right.
Yeah. It's like we're coming in at a bit of an angle. But
Yeah, so if you kind of bring your heading around to the left a bit to port,
not to
as you lateral over, if you
okay.
like square yourself up to the slope,
Okay.
it'll be easier to land. And then get it kind of right in front of the lasers.
Yeah.
And then when you try to land, you kind of go down gently And then once you kind of hit bottom, you can kind of give yourself a little bit more down until you're stable. It's hard to tell because you can't see here, but I can put your camera down because you see, so you can see the porch.
There we go. Okay,
Yeah.
thank you.
And keep going down.
Right,
You probably want to give like
coming
maybe
down.
30–40% on the verts down to hold there
Okay.
until you're kind of stable. Kind of try to keep it constant, the verts.
All right.
That's pretty good.
She's
And then
coming down.
then when you feel stable, you can hit stick lock.
Okay. Turn on that. Am I on the ground?
Yeah.
Am I landed yet?
Yeah, you're landed.
Am I?
Yep.
Okay, and then stick
Stick
lock.
lock, I can get it for you. Yeah,
There you go.
nice.
I think the top, if I remember right, is stick lock as
Yeah, that's
well.
also stick lock, yeah.
Okay. All right, you can zoom in on that.
Yeah, I'll tilt back up a bit. Nice job.
I guess this would be the time to swap out.
No, we can wait for Dan and to come up. Are we sampling this? I didn't hear.
They haven't
No? They
looked
just
at
wanted
it
to.
yet.
They just wanted to look at it.
Just look at it.
Okay.
Okay. Right.
We're having a shift change here, and so there's going to be a few moments of quiet here while people get settled and oriented.
You can come wide, Amber. Thanks. Oh, yeah.
Here's a fish.
This might be the same one we saw a couple of minutes ago. They're pretty slow.
Bony-eared.
Oh.
Oh. Acanthonis armatus.
I get out back ahead. Jaden, can you come up on the winch like 5 meters as I come under?
I gotta step off just for a moment there, Jake. I'll be right back.
Yeah. All right, no worries. Yeah, Dan's coming up. He said he completely lost track of time.
He was sitting down there playing with his toys.
Yeah.
You want to start the ship moving?
Sure. Let's do it.
Bridge, nav, 50 meters, due north.
You can bring Atalanta's heading around too, Jaden. To, I think 330 or something. Oh, due north. You're going due north?
Due north.
Due north.
To the north, onward.
But first, let's zoom fish.
Want to push in here on this fish, Amber?
Get the net out.
Second, guys. Jeez. Okay, ready. Are you ready?
Yep.
Okay. Don't run away.
I dusted it up.
So what was that, Harold?
This looks like a cusk eel.
You
Possibly
combine-
the same one we've been seeing quite a bit on the Stive, but didn't get too good of a look at it. The head shape looked maybe a little more
Right,
flat
if these aren't going to work, I'm
than
going to go back to this one.
Bassizetus, but I don't know. We've got Jeff watching in the lounge, so waiting for him to chime in.
I want to thank Chris Krasnowski again for turning on the ultra high resolution side scanned sonar that's doing such an extraordinary job, and that shows we're on this featureless plane continues to our right and to our left for some distance.
You can see the complete lack of features in this terrain with that thing.
Well, and Chris, I want to also point out that this is so accurate, as I asked you a moment ago, that you would be able to see, we would be able to see a single shell casing like the one we saw earlier.
That's correct, especially in the backscatter, so in the coloring of the points there, they'd show up.
And we would pick up debris from a shipwreck from World War II.
Yeah, potentially.
Yeah, Chris, so the coloration is backscatter and the shape is bathy?
That's right.
Okay.
I had the backscatter waterfall up earlier, but
I got out ahead.
So for our viewers, they're talking about the view you can see on satellite feed 3, channel 3, in the bottom right corner.
See?
Is that the side scan sonar?
Yep.
Terrific.
Yep.
Bridge, did you put in that move? Am I just looking at the wrong screen? Did you put in the last move? Which DP are you using? Port or starboard? There we go. Thank you. Oh, nice for you to join us, Dan.
Bad noise.
All right, Ali, we got watch for 4 to 8 today, or right now this afternoon.
Wow, I feel like I've stepped back into 1999.
Are ROV upper and lower cameras off intentionally?
ROV upper and lower cameras are off. We have, I'm pulling you. We're having some network issues. We're trying to, our top scientists are all over it as we speak.
Copy, awesome.
Weren't you just bragging last watch about how you fixed the network issues?
Yeah. But I believe I had a caveat in there.
I'm sure you did.
All right, is it a good time to get have some introductions?
Sure.
All right, let's start with the back row.
Oh, good afternoon ship time. Good early morning North America time. My name is Brian Kennedy. I'm the Chief Scientist at the Ocean Discovery League. I'm also the External Science Advisor for NOAA's Beyond the Blue Pacific Islands Exploration Campaign. I'm a deep-sea benthic ecologist specializing in the ecology of seamounts, and I am the watch lead.
Ali, everybody. My name is Harold Carlson. I'm a PhD student at the University of Hawaii at Manoa. I study deep-sea ecology as well, specifically fishes and their habitats, like deep-sea coral and sponge gardens. And I'm in the science chair for the 4 to 8 watch.
Ali, and hello everyone. My name is Ayla. I work over at the Coral Reef Research Foundation in Palau as a Marine Lakes Research Assistant, and I'll be the data logger for this 4 to 8 watch.
Ali, my name is Elle, and I'll be the Science Communications Fellow on this watch from 4 to 8, and I'll pass it on to our special guest here in the back row.
Thank you, Elle. This is former Congressman John Culberson from Houston, Texas. I'm delighted to be here. This is my 9th expedition on the Nautilus. Now that I'm out, retired from Congress, I'm a member of the board of the Ocean Exploration Trust. And in my time in Congress, it was my privilege to serve on the Appropriations Committee, where I worked in support of a lot of local wonderful transportation projects in Houston, but in particular the support of the sciences, NASA, the space program, ocean exploration. I was actively involved in launching the Europa Clipper mission to the ocean world of Europa, and it was my privilege to help support ocean exploration in my time in Congress on the Appropriations Committee.
Can we catch him?
We as much as you might like to, Dan, we already have one, I'm pretty sure.
Oh.
Yep, we already caught this one.
Sounds like one of the other watches was able to catch the fish with the net.
With the net?
This was a slurp.
Oh, all right.
Yep.
So we still haven't netted a fish?
Still not netted a fish.
Still not netted a fish. If we see a rat tail that's too big to slurp, but not too big, I think that might be our target.
Roger.
But let's take a quick zoom on this while we're here waiting for some leash.
This is Chonocops, I believe?
No, this is
what Megan
it's
said,
a type of
or
batfish.
Batfish, okay.
Why are we going downwind? Can you get the current to go the other way?
Hey, at least there's a current now.
The fish is telling you which way the current's
So
facing.
this is a pretty interesting, this is the
Go ahead,
second
Amber.
one
Push in halfway.
we've seen that's just doing this floating. This family is normally very benthic. It sits on the bottom, even tends to walk a little bit, and I've only seen them swim when extremely perturbed in other parts of the world. But this species, which is new to me, I've never seen this one before. This is 2 individuals now on this dive that have been just hovering half a meter or so above the seafloor, which is new behavior for me.
Jeff says it's solocisquama.
How big do they get?
This is probably full grown. The other members of the same family are all about this size.
Is there a common name for this fish?
I don't know about this one specifically. The
us.
family I've generally called batfish. And you can find these all the way up
That's
into
pretty
scuba
cool.
diving
That
depths.
is
Nice.
a great close-up.
All right, thank you, Dan.
That is definitely going to be a highlight.
Yeah, I don't know about that. Need 60 seconds. 60 good seconds. I don't know. Our production team is pretty good at those shorter clips.
5-0 meters.
They always yell at me if I don't have 60 seconds of good video for them to edit.
The largest species in this genus only gets to about 14 centimeters in length. So pretty small.
We got a couple Comanche stalked crinoids and looks like maybe another smallish Ritogorgia. We were having an interesting discussion, Harold, Jeff, and I down in the lounge on the last watch when we were watching the dive about the distribution of size classes of corals and how there's been very little studies done on basically the distribution of size of different types of corals in a community. And the reason we're interested in the different sizes is it can give us some understanding of their reproduction and recruitment. Are there, is there a wide range of different sizes all at the same place, or does the same place have all the same size corals, and you can kind of roughly think size as an analog for age? So the fact that we've seen a couple smaller Ritogorgias on this dive, just an interesting kind of thought of why you might have a recruitment event for Ritogorgias on this seamount at a certain time period. I'll be interested to see as we get higher if we end up finding larger ones, or if we continue to see this kind of smaller sub half a meter size class continue to be the dominant size class for the genus.
Push in there a bit for me.
Copy.
Go halfway there.
Quite a few of these swimming sea cucumbers on this dive. And despite it being lots of flat sandy bottom, we did go through some rocky areas, but this sediment zone seems to be very active. You can tell from the amount of bioturbation in the sediment, just those little hills and mounds and trails and patches of activity. It shows that there's quite a lot of life digging around.
You want to go a little bit deeper and explain bioturbation and some of its importance for the ecology?
Here's another rat tail. Bioturbation is basically just organisms stirring up the sediment and moving nutrients and carbon from layer to layer and mixing things around. And you can see all of these little mounds and holes. Those are all created by animals, and that's what we refer to as bioturbation.
Like having lots of earthworms in the soil.
Exactly. Yeah, if we could get a zoom on this. Well, we've got an interesting sea star and a fish.
You want the fish or the sea star?
Well, the fish might get away, so let's try and get a zoom.
Go ahead, Everett.
It's impressive to see the side scan sonar even identify little like sand dunes or mud dunes.
Yeah, really good high resolution sonar is amazing.
Extraordinary.
One of the kind of